All right, hello everyone. We are here to work on the proof of enzyme action lab, and so we're really just trying to see if the enzyme that's in our saliva actually works on breaking down starch, which is a polysaccharide, into glucose, which is a monosaccharide. So that's our goal. Again, what I have here, just so that you're aware of the materials, is I have some Benedict solution, which is that blue stuff that in heat turns to sort of a rust color. I also have iodine, which doesn't need heat, but it turns from that amber color into more of a black or purple color in the presence of a polysaccharide. The Benedict's was for a monosaccharide. And so then I have 6 test tubes that I'm going to be using today, and I have my starch substance, which is actually just a saltine cracker, and we'll get more into what that's going to be used for in just a minute. If you're in class for safety reasons, we have to be careful because we do have a beaker of water that's on a hot plate, so we want to be careful with that. And also just handling glassware, and when we're heating stuff up, we would always point the glassware away from us. So just different things like that we want to be careful of handling stuff that's hot, and also just the glassware. Because I am using chemicals, again, my main safety thing is I will have my goggles on, so we'll go ahead and put those on. And just to get started, I need to crumble up some of my cracker here, so just so I can have some little bits of it. And I'm going to put 2 test tubes. I'm going to have a little bit of crumbled cracker up in, just so it barely coats the bottom of the test tube, just a little bit in there. And I'm going to have 2 test tubes of that. And then I have to have just plain old saliva in 2 different test tubes, so I need to kind of spit in 2 of them. So again, kind of the gross factor here. Okay, and like I say, again, just enough so it's at the very bottom of the test tube. And then to save time, I went ahead and chewed up some cracker ahead of time, but this takes about 5 minutes to chew up the cracker. It has to be so it's kind of like cracker mouthwash is how I describe it, but it can't have any chunks in it, because you got to be able to suck it up into like an eyedropper and drop it down into the test tube. And so we're going to do that. Just again, getting it down there in the bottom of the test tube. And then if you recall, it takes about 5 minutes for the Benedict's test to work. And so I'm going to go ahead and get that one started, and then we'll do the iodine test while we're waiting for it to work. And so again, for Benedict's, I need 2 mLs of my Benedict solution, which is basically a dropper full. And so I'm going to get 2 dropper fulls. I'm going to put 1 in just my cracker. And then I'm going to use just my saliva. And then 1 that has chewed up cracker. And I can put my Benedict's off to the side. And so I'm going to take those and I'm going to keep them in that same order. And I'm going to put them in my hot water bath and just let them sit there for about the 5 minutes, and we'll see if anything happens when we're kind of done with the iodine test. For the iodine test, again, I already have my cracker and everything here, and so I'm just going to kind of put my iodine in there. Remember, that just takes 2 drops. And for the 2 drops, I'm just looking at it to turn from that amber color into like a dark black or a purple. And so, I don't know how well you can see that here, but you can see there's a definite black on the bottom of just my cracker. And so that's my first result for iodine test. For just the cracker, it was black. If you can see the saliva there at the bottom, it's really just still amber. There's no color change on that one, so that one's just amber. And then for my chewed up cracker, you can see hopefully you can see that there's black in there as well. Okay, and so that one is also black. Okay, and so from there you should be able to tell me, remember that's testing for a polysaccharide, and so you should be able to tell me which one of those down here are polysaccharide, and then, or monosaccharide or none. Actually, I should put up here as well, it is possible to have both as well, so you might want to add that to your lab sheet as well. But that is a possibility also. Okay, and so that's that one. Let me go ahead and look at our Benedict's test here real quick. We're just now starting to get a little bit of a change, so I'm going to let that go a little bit longer. Let me mix them up just a little bit here. They are a little bit warm, but... Okay, and again, I'm being careful to keep them in the same order. Okay, but you can, actually we can kind of pull those out right now. Move these 3 test tubes over. It didn't go the full time, so I'm not going to see the full result, but you'll get the idea here. So that's my chewed up cracker. There's my saliva. And this is my, that's actually my chewed up cracker. This is my just crumbled up cracker. So for my crumbled up cracker, you can hopefully see that's still just blue. And so let me put that on the board. For my saliva, you can see that's also just still blue. But now for my chewed up cracker, you can sort of see it's turning, it's at the green stage. Remember I told you there's phases as it goes along in terms of this Benedict's test. It starts to turn green, then it turns kind of a yellowy orange color, then it finally gets to that rust color at the end. So if we would have let that go a little bit longer, it would have gone to that rust color. But you can, right now it looks kind of like pea soup if you want to kind of look at that color there. But anyway, we're going to put rust for that one just because of the fact that it would have gone all the way to the end if we would have let it go longer. Okay? And so now from these results, I should be able to tell if there's just polysaccharide, monosaccharide, none, or both for that. Same for my saliva, and same for my chewed up cracker. And so that's your job, is to fill in those last 3 blanks, and then also fill in the other ones as well. And then you have some questions to answer and a conclusion to write that is in the CER format. Just a reminder, on the CER format, if you don't remember how to do the CER format, you can go back to one of those purple buttons on the home page for the class, and the lab report format sheet is found there, and you can go ahead and refresh your memory on how to write the CER conclusion. If you have any questions, just let me know, and I'll be happy to answer those as well. But that is it for the Proof of Enzyme Action Lab, and hopefully you do well, and just don't forget to hit submit when you're done. Bye.